Hi everyone, this is Jeroen with Phospho for Entagma, and today we're going to look at how to visualize molecular structures of proteins in Houdini. In particular, we're going to be looking at the coronavirus and then the coronavirus spike protein. So before we do that, let's have a look at what the coronavirus looks like. So you'll have seen this image made by Alyssa Eckert and Dan Higgins at the CDC. And what you're looking at here in gray is the membrane envelope of the coronavirus. Inside is all the genetic material that the coronavirus then pumps into healthy cells to infect them. On the outside you see in yellow and in orange are the envelope and membrane proteins. They're important for the structure of the virus. And then in red are the spike proteins or S proteins. And these are really important because they are the proteins that are binding to the receptors on the host cell, and that initiates the infection of the host cell. And the way that they're arranged here with their shape, it makes it kind of look like a crown, and that's why they're called coronaviruses. If we want to animate what these look like, first we need to get some scientific source material. Here's a paper that came out recently by David Wiesler and his lab in Seattle, and they've solved the structure for the coronavirus spike protein, but they haven't just solved a single structure, they solved two. So we've got on the top here the closed coronavirus spike protein, and at the bottom a partially open one. And that's really important because in that partially open state, we've got this protrusion here, and that's the thing that's actually making that interaction with the host cell receptor. Now you see that these have three colors, and that's because this is a trimer. What that means is that it's not a single protein, it's one protein with three copies that are bound together in a complex. So you've got in blue and in pink and in yellow, it's the same protein, but there's just three of them combined together. But because we have this closed and this partially open conformation, we can then put that into Houdini and have a look at animating that structural change, which we call a conformational change. So in order to do that, first we need to get our hands on the scientific data behind this. And all of the protein structure data that you ever want is collected in the protein data bank. So this is an absolutely enormous resource full of all of the different protein structures that people have solved in the past. And there's a very helpful link here to coronavirus specific features. And we can then scroll down and find spike protein PDB structures. And if we scroll down, it's 6VXX and 6VYB that are from that paper that I was just showing you. If we go to 6VXX, download files, and make sure you get the PDB format because that is natively read by Houdini. So if we hop over to Houdini, drop down a file, go inside, and in this file, let's read out 6VXX, and we'll call this closed. Drop down another file, and we'll call this, let's get 6VYB, and we'll call this open. If we hit F, we can see that here we have our structure, and it imports as a point cloud where each of these points represents a single atom in that structure. So if we have a look at the geometry spreadsheet, you can see that there's a lot of information. So obviously we get our atomic coordinates or our point position. Then there is the chain ID. I just told you that this isn't a single protein, but there's actually three copies of the same protein. And so in this case, each of these copies has a particular chain ID. So we've got chain A, we've got chain B, and we've got chain C. What we're going to be doing today is we're going to visualize each of these three chains in a slightly different way because there are several ways that scientists like to visualize proteins, and we're just going to cover our bases on that. Then each of these atoms in the point cloud has its own element, of course. So there's a nitrogen there and a carbon, and oxygen, etc. And then because there's so many different carbons in there, if you look at the name attribute, there's a little bit more information about exactly which carbon it is in the arrangement. If we look here, the two important ones to look at are res name and res sec. So it's the name and the sequence of residues. Now a protein is a chain built up out of amino acids, and there's 20 different amino acids, and they're oriented or that they're arranged in a particular order, and that gives you your protein. So in this case, at sequence number 27, they don't always start at number 1, that's fine. So at sequence number 27, we have an alanine. At sequence number 28, there's a tyrosine. At 29, there's a threonine, etc., etc. And the last thing that we're going to be interested in here is the temperature factor. Now, each of these atoms in our structure has this temperature factor, which is a measure of kind of how flexible they are in 3D space. And we're going to be using that to create some procedural noise animation. But as you can see here, our protein structure is sat somewhere in space, and we'd like things to be at the origin just to make it a little bit easier to work with. So let's pop down a transform, move it to the origin, and let's change this around a little bit to these values, and let's copy this across for the open conformation. If you go back and forth, you see that the open conformation is rotated slightly compared to the closed conformation. Let's get rid of that difference because we want them to be as close to one another as we can get them. So something like this works well for me. And they're now oriented flat on the ZX plane, and let's have them in their actual orientation. So if we rotate them minus 90 degrees, they're upright, and that's the way we like to be looking at them. So one of the things with these protein structures is that the coronavirus spike protein is covered in sugars called glycans. We don't want to have these sugars in this structure anymore because there are differences between the closed and the open conformations in where those glycans are, and we're going to be interpolating between the two structures, and it's just a little bit easier if those glycans aren't there anymore. So to get rid of them, let's drop down a wrangle, and in our geometry spreadsheet, if we look at the residue name and we scroll all the way down, there's this NEG here, and those are the points belonging to those sugars, and those are the ones that we're going to have to get rid of. So let's say if our string attribute res name equals NAG, then let's remove points over stream 0, and let's remove the currently evaluated points at ptnum. And if we middle mouse click on the transform 3 here, we see that we have 23,700 points, and here we have 22,800 points. So we've gotten rid of quite a few. Let's call this remove sugars, and do the same for the open stream. I mentioned before that we're going to be visualizing each of the 3 chains in a slightly different way. So what we're going to do is we're going to group out each of these chains, and then we can isolate each one separately. So put another wrangle. Let's call this one group chains, and go at group chain A equals string at chain ID equals A. And we can copy this across for B and C as well, and do the same for the open stream. And to keep things a little bit nice and tidy, let's put down a null and call this closed start, and another null for open start. So we can put down a blast here, and in this blast, what we want to do is look for chain A and delete everything that's not selected. And now we've isolated chain A for both the closed and for the open conformation. So to morph between them, we can just put down a blend shapes, pipe in both, have a look at that, and do our blend shapes. The problem here is that it's not working. So what's happening here is that the number of points for the closed conformation and the open conformation isn't the same. If we look, we have 7,600 points here, and we have 7,400 points here. And what the blend shapes is doing is that it's looking at the point position for point 1 in this stream and morphing it to the point position of point 1 in this stream. But if the points don't match up, then it's going to drag them all the way across to the other end of the protein. So that's not what we want to do. So we need to match up those streams. In order to do that, let's get rid of this blend shape, and let's put down another wrangle. Pipe our closed conformation in the first input slot, our open conformation in the second, and first of all put down an integer matching at 0, and then we need an integer ressec, and that's pointing to the ressec attribute. We'll then want to count how many entries there are for each amino acid. We need an integer ressec count 1, because we'll be doing this for both of them, and that's find attrib value count over stream 0. We want to look at points, we want to look at ressec, and we'll be evaluating the attribute ressec. We can now copy this across, do the same for 2, except in this case, we need a comma here, and in this case we will be looking at the input stream 1. So if ressec count 1 is the same as ressec count 2, everything is matching up nicely, so matching is 1. So let's write out matching as an attribute, and let's write out ressec count 1 as an attribute as well. And the same for ressec count 2, so we can have a look at what's going on. And we can then group a no match group where the integer at matching is equal to 0. So let's have a look over in our geometry spreadsheet. We sort by matching. Kidding me, that was working before. You're probably looking at your screen shouting at me, no, there's a typo. Oh, there it is. So I missed out that one. And there was a typo here where in our if clause, if ressec count, it needs 2 equal signs. Okay, so now if we look at our geometry spreadsheet and if we sort by matching, we can see that there's 2 scenarios here. In the first case, we've got the full 8 atoms in our amino acid here for count 1, but we've got nothing for count 2. So basically, in this case, the entire amino acid is missing from that structure. In that case, we want to get rid of everything. But there's also this case where we've got 5 atoms left in this particular amino acid, but the other one has 8. It turns out that those 5 are always the same and they're always the backbone. So I checked and they're always this N, Ca, Co, Cb. What we're going to do, instead of getting rid of the entire thing here, is from resset count 1, we're just going to keep the ones that we also have in resset count 2. Now before we do that, we need to make sure that we copy this one across and change across the inputs to do it for the open stream. So let's add another wrangle. Because we now have our no match group, what we want to do is we want to take those bits that are still interesting to us and we want to remove them from the no match group. And then afterwards, we have our proper no match group that we then can get rid of. So first of all, if the resec count 2 is bigger than 0, because if it's 0, then we need to get rid of everything. So we're not interested in those. If it's bigger than 0, then another if, the string at name equals n, or if the string at name equals ca, or if the string at name equals cb, or if the string at name equals c, or if the string at name equals o. These are the ones that we want to keep. So in those cases, let's say set point group over stream 0. We want to look at the no match group. We want to look at the currently evaluated point at ptnum, and we're going to go with 0 to indicate that it's no longer a member of that group. If we middle mouse click on the wrangle here, the no match group has over 500 points in it. And if we middle mouse click here, it now has 280 points. So we've saved over 200 points that are relevant to us. Cool. So now we can just blast away the entire no match group. And if we take these last 2 nodes and we copy them across for our open confirmation, hopefully by middle mouse clicking, we've got 7,323 points in the closed confirmation. And we also have 7,323 points in our open confirmation. So now our blend shapes should work. Hopefully, fingers crossed. And indeed it does. We see some nice subtle conformational change happening there. But before we do our blend shape, let's add a bit of procedural animation to it. So I mentioned before that we have that temperature factor that's a measure of how flexible each of these points is in space. So let's use that as a multiplier on some curl noise to give it a bit of a wiggle in space in a way that's driven by the scientific data. The problem though is that that temperature factor is not particularly in a range that we like to work with. So let's normalize that first. And in order to do so, we're going to add 2 attribute promotes, and we're going to look at the temp factor, which is a point attribute. We're going to promote that to detail, and we're going to promote the maximum. We change the name to temp max, and make sure that we don't delete the original. And we'll do the same here, except this time we're going to be looking at the min. Fantastic. We can now add another wrangle. We'll call this normalize temp factor. We want to have a float min that is detail over stream 0. The detail attribute is called temp min, and copy that for a float max, which is looking at temp max. We now want our float attribute temp norm for temp normalized, and we'll use the fit looking at temp factor. We're going to normalize it by min and max, and we're going to fit it to 0 and 0.1. No, sorry, 1.0. And if we have a look, now we have our normalized temperature value, and that's in a range from 0 to 1, so we can work with that. It's not normalized, it's normalize. So then we can put down a point fob, call this temp noise, put down a curl noise, put our P into the position there. We need to bind our normalized temperature, so our temp norm, add a multiply. We need to multiply the noise with our normalized temperature, and we can then add that noise onto our position and put the position back into P. So let's promote some of these parameters so we can control the noise 1 level up. Okay, and so the frequency we're going to keep at 1, the amplitude is going to be 0.5, the roughness will keep the same, attenuation of 2, turbulence of 4, and then we'll animate the offset. So let's go from 0 to 1 at frame 240. And we alt click to set a keyframe. And if we go into the animation editor, we want to make sure that we get rid of that smoothing. And so we have a linear animation there. If we now press play, then we can see that there's some wiggle happening here at the top, but not at the bottom, and that's because that's a much more rigid part of the protein. Let's zoom in a little bit. So we've got flexibility here and less so in the more rigid regions. Fantastic. So in doing all of this, we've created a bunch of attributes that we no longer need. So let's tidy things up, put down an attribute delete, and let's delete everything except for P and then all of the attributes that were in the original data. So res in name, res sec, we want to keep the temp factor, we want to keep the elements, the chain ID and the name. Now we are ready to do our blend shape. So we put down a blend shape. So we now have our nice interpolation and press play and you can see that we've got our noise from the temperature factor. But we're not going to visualize this as a point cloud. We want to, in this first of the three visualization types, we want to use a surface rendering. So we'll use a VDB from particles for that. And this we want to keep relatively coarse. So we'll have a voxel size of 1.25. We'll have a point radius scale of 2.5. And we'll have a minimum radius. We'll keep that 1.5. But this needs to be smoothed out a little bit. So let's add a VDB smooth SDF. And let's keep it at 2 iterations to keep some of that detail. We can now convert the VDB back to polygons, and let's add some normals to this. So one of the things that we want to do is put down an attribute blur. The reason for this is that when you've got conformational changes happening, sometimes your geometry can go a little bit jittery when those changes are happening. And so we just want to blur that out a little bit by blurring P and the normals as well and putting down 2 blur iterations. So that's it for this, except the fact that let's put down a color so we can see the differences a little bit better. And if we want to stick to the coloring scheme that they used in the paper, then that chain A was yellow, and let's go for a yellow somewhere like this. Okay, save. And for the second chain, we can reuse quite a lot of this work. So everything from the blaster all the way down to the blend shape, that's going to be the same. So let's copy all of that across. But of course, we need to make sure that we're not isolating chain A, but we're isolating chain B. Let's go down to our blend shape and visualize it here. Okay, so for this one, what we're going to do here is we're going to look at a ribbon diagram of the protein. So for a ribbon diagram, what we need to do is have a look at the geometry spreadsheet. And the ribbon diagram is basically connecting all the backbone carbon atoms. So that's the Ca, the alpha carbon. We need to connect all those together. So basically we need to group our alpha carbon and we need to get rid of everything else. So let's put down a wrangle. Let's say at group backbone. And if the string at name is the same as CA, then it'll be in that backbone group. We can then blast everything that's not in the backbone group, and here are all of our backbone atoms. We then put down an add sop, we go to polygons by group, and now all of a sudden we have our chain connected. Because this is a little bit jaggedy, we want to add a subdivide just to smooth things out a little bit. And the nice thing about using a subdivide here instead of a resample node is that you're still basing things on the original points from the data file, and so that's a little bit more scientifically accurate than if you do a resample and it's slightly moving things around a little bit as well. So we've got things subdivided, and what you would say in order to get a ribbon diagram is to add a polywire, pipe that in, have a look. It's a little bit thin. Let's go to say 2, add a bit more division so we can see what we're looking at. But if we press play and you have a look at the topology here, then we see some really annoying, weird rotational artifacts happening. So one way to get rid of those is to dive into a whole lot of quaternion math, and I'd like to avoid doing that. So let's get rid of this polywire and let's instead use vellum. Having said that, if you're just doing an illustration, going down the polywire road is absolutely fine. But for us here, let's do our workaround and let's first put down a group. This will be a point group, and let's call this pin, and we want to put everything in there. And I just realized that we've not animated our conformational change. So sorry about that. Let's go back to our blend shape here, our first blend shape, and visualize it. And let's say at point 72, it's the first shape. At frame 120, it's the second shape. Alt-click to set a keyframe, and now we've got a conformational change. Let's do the same for the ribbon motif. So at frame 72, it's keyframe at 0, and then it's a frame 120. Keyframe that at 1. And if we visualize this, we can see that we now have our conformational change happening. Great. So we've got everything in our pin group, and let's put down a vellum configure hair. What we want to do here is we want to, in our pin to animation, select our pin group, set the pin type to soft, and match the animation. We also want to go down and increase the bend stiffness to as high as it goes. Then we put down a vellum solver, plug in the first and the third slots, add a wrangle, And so in this wrangle, we plug the constraint stream. We want to let this run over app type equals pin orient. We want to run this over primitives, and we want to remove the prims. So that should get rid of our weird rotation artifacts. We put that into the constraint geometry, and in our solver, we're going to pull our constraint iterations way down to 1, no smoothing iterations, no collision passes, no self collisions, and just turn off the collisions as a whole. Highlight the Vellum solver. Let's save this and play. Okay, so that still seems to be behaving the way it should. The Vellum solver creates an awful lot of point attributes that we no longer need. So let's put down an attribute delete and tidy things up a little bit. We want to get rid of everything except for the orient and resec attributes. Let's subdivide this because it can be a little bit smoother, and put down a sweep with a circle set to polygons. So I like these to be a little bit chunky, so let's increase the scale of that circle, and we want some end caps, and this is inside out, so let's reverse it. If we now press play, then you can see that our geometry is much more stable than it was before, and much less of that weird rotational artifacty stuff happening. Great, so the last thing to do here is to give it a color, and I think in the paper chain B was blue, so let's go for something this. Okay, that's our second chain done. For the third chain, we're going to be looking at a much more detailed version of the surface, and so we're looking to show some of the individual side chains of each of the amino acids. So in order to do that, we basically need to just use that same tree that we've done here for chain A and just play with the settings a little bit. So let's copy this entire tree and let's paste the tree over here on the right. We obviously want to make sure that we're not evaluating chain A anymore, but we're going to be looking at chain C. Okay, so we're going down to our blend shapes to have a look, and this looks like it's still behaving properly. In the VDB for particles, this is way too coarse for us here. So what we'll be doing is let's take the voxel size down to 0.5. Let's take the point radius scale down to 1 and the minimum radius to 1 as well. In the VDB smooth, let's only do a single iteration. And when we look at the converted polygon here, you can see that there's a lot more detail there. And so now you can clearly see the individual side chains of each of the amino acids. That's giving us a lot of lovely detail. Same as before, we add a normal, we blur some of the attributes, particularly the P and the N, to make sure that everything looks a little bit nicer when animated. And in terms of a color, I think this was a bit pink in the paper, so let's go for something like this. So now we've got our 3 different trees, where in each of the trees we look at one of the chains of the coronavirus spike protein. We visualize them in 3 different ways. So now we can bring that back together with a merge, take our surface view for chain A, take our ribbon view for chain B, and take our individual amino acid view for chain C, and we need to add a normal to our ribbon view. Okay, so here we've got the entire coronavirus spike protein visualized in Houdini, and let's see how it moves by zooming out a little bit and doing a flipbook. So, to recap, we've got the coronavirus spike protein here. We've visualized it in a surface representation in yellow, in a ribbon representation in blue, and then in an individual amino acid residue representation here in pink. There's some really nice structural features that you can see in what we've done here. And so, for instance, some of those dimples in the structure in yellow in the surface representation, you can see that there are actually some amino acids from one of the other proteins that are poking in there to fill that space. So you can really see how that structure binds together in the trimer. The other thing we've done, if we press play, as you can see, we added a bit of wiggle based on the temperature factor, and that wiggle is predominantly happening in the areas that are more exposed of the protein. And then if you look down here where everything is bound really tightly together, there's hardly any of that wiggle happening at all. And then of course we have our conformational change where we go from a closed state to a partially open state. You see that here in blue. And the nice thing is here that on the top are the contacts for contacting the receptors in the host cell. And what you can see here is that those contacts are buried and inaccessible in the closed conformation, and you go to the open and they're accessible and ready to bind to the host cell and start infection. So it's really great that Houdini is able to directly import these PDB files as atomic point clouds, including all of the different information that the scientists put into those files. And you can really make sure that you're creating a scientifically accurate animation that's all based on the underlying scientific data. So that's it for me. Thanks very much. You can find us on Twitter at Phospho. You can find us on YouTube if you look for Phospho Biomedical Animation. And thanks for watching. Cheers. Bye-bye.